Level 4 Biology, Cell Biology. In this video, we are going to explore the properties of enzymes and their use in industries. Our learning intention for this video is to develop an understanding of the properties of enzymes and their use in industries. Page 15 of your pupil booklet will identify the success criteria for this particular topic. We've covered the learning in the previous video, but we will be revisiting the function of enzymes and the properties of enzymes, as well as the reactions and an application of them in biotechnology industries during this video. Before we progress with the content of this video and the practical investigation that you're going to carry out, you have the opportunity to watch a summary video from BBC Bitesize with regards enzymes. The link is shown on the screen in front of you. Please take a moment to copy the link, watch the video, and come back to this tutorial once you've finished. In terms of an enzyme recap from our previous lesson, we learned that enzymes are biological catalysts that speed up the rate of a chemical reaction. An important aspect of an enzyme is that it remains unchanged by this reaction. That means it could be used over and over again. Enzymes are specific, meaning that each type of enzyme will only react with one type of substance. The substance that an enzyme is specific with is called its substrate. Enzymes speed up the breakdown of complex molecules into simpler ones, or they can be involved in building up simple molecules into more complex ones. From page 18 of your pupil booklet and in class, you will have the opportunity to carry out a practical investigation. The aim of the investigation is to investigate the catalase concentration of different tissue types. We can measure the activity of an enzyme by measuring how much oxygen is given off during the catalase reaction. Oxygen is a product of catalase acting upon hydrogen peroxide. The tissues that you will use in class, for example, liver, potato, carrot or celery, contain catalase. When they react with hydrogen peroxide, oxygen will be produced. We can measure the quantity of oxygen that's given off by the level of bubbles of foam that appear on the surface of the liquid. Therefore, by measuring the height of foam using a ruler, we can identify the rate of a reaction. The higher the level of foam, the more catalase the tissue must have contained. On page 18, you have instructions and a method for carrying out this investigation. You need to leave the tissue for 5 minutes, and at the end of the 5 minutes, you measure the height of foam. On page 18 of your pupil booklet, there is room for you to complete a results table, identifying the type of tissue you used and recording the height of foam that was produced as a result of the reaction. There is also space on page 18 for you to complete a conclusion, thinking about, did all the tissues have the same catalase concentration? Which tissue contained the highest catalase concentration? And identifying which tissue contained the lowest catalase concentration. You will find tasks on page 19 of your pupil booklet to be completed, identifying examples of properties and functions of enzymes. Effect of temperature on enzyme activity. Temperature has an effect on enzyme activity. The graph that we can see on the screen shows the effect that of an increasing temperature has on the rate of enzyme activity. At low temperatures, there is a low rate of enzyme activity. As the temperature is increased, so as we go from left to right in the bottom of the graph, we can also see that the slope of the graph is increasing. So as temperature increases, so does the rate of an enzyme activity. You will notice from the graph that there's a peak, and then the enzyme activity starts to decrease. This maximum activity is what we call the optimum, and we would identify the temperature as being the optimum temperature for this enzyme. That means the temperature at which the enzyme is most effective or most active. You have an activity on page 20 where you've been asked to reflect upon the effect of temperature on enzyme activity. You've been asked to reflect upon the differentiated success criteria and gauge your judgment of your own learning. If there's any of these success criteria that you're still struggling with, please go back to the relevant point of this video or our previous video and make sure you're revisiting the learning. As part of our plenary, you're asked to answer the following two questions. Question 1. Identify from the diagram below the products, the substrate, and the enzyme. For question 2, explain the action of an enzyme in a chemical reaction.